Hi everyone, I want this video to go over the nephron loop diagram. You can see that this is a PDF drawing that I have posted in your Canvas page, but I'll also be talking through it, redrawing it, talking through it, adding some other clarification points in this video. The nephron loop is typically one of the most complicated processes that you will encounter when granted, once that it's kind of explained, I think it's pretty straightforward, but there's a lot of details to it. And intention of this video is to walk you through those steps, walk you through those details, and understand those foundational or fundamental concepts that will help you understand it. While this is probably one of the most complicated or complex processes in this chapter that you'll encounter for this exam, it is not necessarily the most important. And since there's not a lot of questions I can ask, but by understanding the nephron loop, it will sort of prove to you that you understand a lot of the other processes and concepts that are occurring in the kidney, because what the nephron loop does is ultimately what you want from the kidney and what you want these nephrons to do, which is to concentrate the urine. So let's go ahead and start off with some fundamental concepts with the nephron loop. There's going to be a lot of detail here that I'm going to throw at you. A lot of it is sort of review, but of course, being a video, you can stop, replay, and go through it and look up things. But you do need to have an awareness of these. I think it ends up being kind of four main topics to understand before we look at the nephron loop and in its entirety. So first is when we look at the nephron loop itself, remember in the nephron, you have the glomerulus, that renal corpuscle, and you have the renal tubules. So you have that glomerulus into the glomerular capsule, and then you actually get into the tubular fluid. So you have the proximal convoluted tubule, you have the descending limb of the nephron loop, you have the ascending limb of the nephron loop, then you have the distal convoluted tubule, and then you get into the collecting duct and on into the rest of the kidney structures, the minor calyx, major calyx, renal pelvis, ureters, bladder, urethra, and so on. Since we're looking at the nephron loop, I want to look first at the kind of anatomy and physiology, this very brief relationship or explanation of the function of the limbs of the nephron loop, because this is fundamental to understanding what the nephron loop does. So first is the descending limb. Due to its anatomical structure, the cells that are there, that epithelium, it is only permeable to water, not to solutes. So of course that means water is the only thing that is allowed to cross the wall of the descending limb. Solute, specifically sodium and chloride that we're going to look at, but no solute can cross that membrane. Then we have the ascending limb. The ascending limb fundamentally is impermeable to everything. Nothing can diffuse across the epithelium of the ascending limb, but it has active pumping channels. So it has sodium and chloride active transport pumps that pump those sodium and chloride ions into the interstitial fluid. We'll talk about this direction of movement a little bit more on the next slide, but you need to know that the ascending limb, impermeable to everything. So water is not allowed to diffuse, solute is not allowed to diffuse, but the ascending limb actively pumps sodium and chloride ions from the tubular fluid, in a sense, from the inside of the nephron loop into the interstitial fluid. So that is to the outside. Then, of course, with talking about the movement of different substances, we need to have a basic understanding of osmosis. Osmosis is just the special name for what we call the passive diffusion of water. So that's what we call osmosis. And just to describe osmosis, what happens with water when it osmotes, or whatever the term would be, is that water moves to the area of the higher solute concentration. So if you have two areas separated by a permeable membrane, permeable to water, not permeable to solute. So if we have it saltier or more, what we'd say is higher osmolarity on one side, so it's saltier, it's more solute, is that water would move from the area of the lower solute concentration, so where there's relatively more water, to the area of the higher solute concentration, to the area where there's relatively less water. So in a sense, always think of it as water likes to reinforce its friends. So if there's an area that, if you think of it like a war, and so you have water versus the solute. If solute has more people in an area compared to water, water is always going to move to reinforce its friends. So if water is losing the battle, we can see water would move to that area where it's losing, where there's less water, higher solute. So this is why the basic phrase that I always remember is water follows salt. Now, salt, of course, if we think of this like table salt, sodium chloride, just because sodium is one of the most abundant ions, and salt being one of the most abundant minerals in your body, but water follows salt. So if we move sodium ion, which we can actively move. Our body has pumps, as we just saw, that can actively move sodium. So if we actively move sodium, water is always going to follow salt based on that principle. Because if you move sodium from area A to area B, that means area B becomes more quote unquote salty or solute or a higher osmolarity. So water is going to follow to that area of higher osmolarity. And of course, I have a little caveat here. This is assuming that the membrane allows water to move, because you could have membranes that don't allow water to move. So if we actively move salt, or this could be actively move glucose, this could be actively move any type of solute, but sodium is the common one, and that certainly is going to be important here for the nephron loop, is that if we move salt, water is going to follow. So whichever way we move salt, water is going to follow. That means water moves passively, and as I said, the solute could be actively pumped in that case, but water still always moves passively. We do not have active pumps for water. Water always moves passively, but solute, salt, sodium, any of these other things could be actively pumped. They also could diffuse. So the last two foundational or fundamental concepts would be the direction of fluid movement related to the kidney. So we've heard these terms before in the cardiovascular chapter, reabsorption and secretion. But of course, it's a little different now that we're talking about the kidney. So when we talk about direction of movement, if we're talking about reabsorption, or your book may use the term absorption, but reabsorption, you need to make sure that you don't get tripped up with this. This is why this is a foundational concept. You need to know that reabsorption is movement of fluid or substance from the tubular fluid into the interstitial fluid, also known as the peritubular fluid. And I'll show an image on the next slide to highlight this. But reabsorption is the tubular fluid into the interstitial fluid or the peritubular fluid, because eventually this goes back into your bloodstream. So fluid and substance that remains in the tubules, if it remains there, you will eventually eliminate through the urine, through urinating. So that's why we say reabsorption is taking fluid or substance that is technically outside of your body, because that's where it's eventually going, and you reabsorb it into that peritubular fluid and eventually back into your bloodstream. So you reabsorb it. Secretion, of course, is the opposite. This is a substance or fluid that is in the interstitial fluid or the peritubular fluid, that area, and then it either diffuses or you actively pump it into the tubular fluid, because remember, we're secreting it. That's going from inside your body, from your bloodstream, through the interstitial fluid, peritubular fluid, pumped into the tubular fluid, because once again, remember, anything that remains in the tubular fluid eventually will be eliminated in the urine. So what's the effects of this movement? And this is where I might lose you, so just pause, take a deep breath. A lot here, and I think even after you watch the video and you come back, this will make sense. So what's the effects of reabsorption and secretion? So let's just do a couple examples here. So if we take sort of area A and area B, and let's call, we could call the tubular fluid area A, and we could call the interstitial fluid, that interstitial space, area B. So if only water leaves an area, so let's say that water is going from the tubular fluid into the interstitial fluid, but we only let water move. We don't let salt, we don't let anything else move. If we only let water leave an area, then that means the volume in that area, area A, will decrease, right? Because you lost water. but the osmolarity will increase because the salt and the solute is not allowed to move. So it stays in the tubular fluid in this example. Water leaves, the salt remains, so the osmolarity in that area will increase. This is the equivalent if you take a pot of water on your stove and you leave it boiling and you forget about it. The water is leaving the pot as steam, but the salt and the solute and the minerals that are in your tap water remain in the pot. And so if enough water leaves, the more you boil it, the more water leaves. So volume decreases because you're losing water in the pot, but the saltiness of the water will increase because those minerals there. And if you really forget and let all the water steam off, you're just going to be left with some white mineral deposit at the bottom of your pot. So let's look at another example. Of course, vice versa too. So if only water leaves an area, then we would say that the volume in area B that the water is moving into, the volume would increase and the osmolarity would decrease. So if only, another example, if only solute leaves an area, so water stays there, this is the equivalent, you couldn't actually do this in real life, but if you have your pot of water and you have some fancy little tweezers and you're able to go into the pot and with your tweezers take out individual ions of sodium or individual molecules of salt. So if only solute leaves an area, so let's say that now solute is going from the tubular fluid and being say actively pumped into the interstitial fluid, so pumped out of an area. So if only solute leaves an area, the volume in that area is going to remain the same because you haven't moved the water, but the osmolarity will decrease because you're removing that solute. So the concentration of solute goes down, which means the osmolarity is decreasing. Once again, that's like your pot of water, and you're able to remove individual molecules of salt from your pot of water. You didn't do anything with the water, so the volume remains the same. It's just going to be less salty, so osmolarity decreases. And then finally, let's say this is just a normal membrane. There's none of these exclusions. If solute is actively transported from our area A into area B, so we move salt from, say, the tubular fluid into the interstitial fluid, and if water is allowed to move as well, so this is just a normal membrane, we actively move salt. Well, we know what happens here. We saw this on the last slide, that if we actively move salt, then water follows salt passively. So if solute is actively transported from an area and water is allowed to move as well, volume in that area A will decrease because we're losing water, but the osmolarity will remain the same because the concentration, the relative concentration of water to solute is going to be the same. Why? Because salt left, so water left with it. So the volume decreased because water left, but ultimately the relative concentration of each stayed the same. So this would be the equivalent of, you've got your pot of water with the salt, and let's say you have 2 cups of water in this pot, and you just take a 1 cup measuring cup and you scoop out 1 cup of water and you dump it into the sink. Well, clearly the volume in the pot, the volume of water in the pot decreased by a cup because that's what you removed, but you didn't change the concentration of the saltiness of the water because the same percentage, the same concentration moved, or you removed it from the pot. So hopefully each of those makes sense. Obviously, if it doesn't, rewatch, think about it, do some research, read about it, and then email me. So let's put this into perspective. You can see the link where I found this image. There's a lot here that's not important, but I think it's just going to be able to highlight exactly what I want to see. We talked about the function of the descending limb and the ascending limb. You can see the descending limb here has H2O with an area leaving because only water is allowed to leave. And then the ascending limb, sodium and chloride, is actively pumped across this ascending limb, but water is not allowed to move. So that's the ascending limb. Most importantly from this image, what I want you to see is that the interstitial fluid, or the what we could call the paratubular fluid, peri means like parallel, on the outside, kind of around. So interstitial fluid or peritubular fluid is out here, whoops. And then the tubular fluid is everything that is yellow here. So remember, reabsorption is tubular fluid into the interstitial fluid. And then secretion is interstitial fluid, peritubular fluid into the tubular fluid. So make sure you keep those directions and those terms straight. So let me switch over to just a whiteboard, and I will go over the nephron loop. And I will more or less be following what I already have listed, what I already have posted in Canvas for you. Like I said, this is important so I can at least talk through it with you and let you hear me talk about what's going on. So first, let's just draw our nephron. So we're going to call this the proximal convoluted tubule or proximal tubule. Then we have the nephron loop. And then I'm just drawing kind of the outline here. So proximal tubule, there's our nephron loop, distal tubule, and then collecting duct. So we're going to call this the PCT. This is the nephron loop. This is the DCT, distal convoluted or just distal tubule. And then this right here is the collecting duct all the way down here. Things might get a little tight here as I'm drawing this. This is why it's important that you have my much nicer hand-drawn PDF already in Canvas. Now, with the nephron loop, this is one of those things, it's sort of like the chicken or the egg. For the whole thing, for the nephron loop to function appropriately, everything needs to be working simultaneously. And so just like with what comes first, the chicken or the egg, it's sort of arbitrary where we start because you don't really know where the best place to start is. And so where I want to, I guess what I want to kind of first establish is that when we are looking in the kidney, right here, kind of across the top, about right here where this dotted line is the difference between the cortex of the kidney and the medulla of the kidney. So the outer region would be across the top here, and the medulla would be the interior. You're getting deeper and deeper into the kidney. And it's important to realize, and then these numbers that I'm putting here are not super important because different books will say slightly different things. And so I don't really care about the specific numbers, but you do at least need to know the relative relationship of each one and what is happening. And I'm actually turning in our book right now just to make sure I can match it as closely to your book as possible. At some point here, I'm sure I will find it. Okay, so looking here, kind of around page 984 is what I'm looking here. But that's why I do have the PDF written for you as well in Canvas. So we start off here, if we're putting some numbers here, we can say that it's like 300, and then we could say, come down here and call it 600. These are osmolarity values. So this is kind of saltiness, as I say. So 600, 900, and then all the way here at the bottom is 1200. But realize this is the same basically everywhere when we're looking in the medulla. So we're going to kind of look at, when we look at the interstitial fluid, I guess I'm saying 300 there. Well, let's look in the interstitial fluid. So we have 300 here, we have 600, we have 900, we have 1200. That'd be the same thing here, 300, 600, 900, and then 1200 down here. So realize that this is the interstitial fluid. It's everywhere around. And so fluid starts off in the tubular fluid, fluid in the tubular fluid starts off at a concentration of 300. So the first question we want to ask is, how does this gradient get formed to begin with? Is what establishes the ever increasing saltiness of this peritubular fluid the deeper you go into the medulla. And so that's where I'm actually going to start. And this is the importance of the ascending limb. So remember, the ascending limb is impermeable to water, but it actively pumps out sodium and chloride ions. So the deeper you are in the medulla, the more sodium and chloride that it pumps. So obviously, the more sodium and chloride that you pump out, the saltier that it will be, the higher the osmolarities. Because remember, you're pumping out sodium and chloride, but water, H2O, is not allowed to cross. So you're moving that sodium and that chloride out. So the peritubular fluid is getting saltier. So the deeper you are, the more sodium and chloride that is pumped out. So as you go up, it's still pumping it out, but it's not pumping out as much. So it's really high down here and it's lower across the top. So that's where you get your little divisions here: 1200, 900, 600, and 300. So then let's see the effects of this as fluid makes its way. As fluid makes its way through the proximal convoluted tubule into the descending limb. Remember that the descending limb is permeable to water, so H2O, check, is allowed to move, but solute is not allowed to move across the wall of the descending limb. So as soon as we get, remember this fluid is like, let's say that it's, just to simplify, let's say that the fluid value, the osmolarity is 300 at this level, but notice what it's being compared to is an osmolarity of 600. Well, what do we learn about osmosis? Water moves to the area of the higher solute concentration. So in this case, water is going to move out into the interstitial fluid, but sodium is not allowed to follow. Solute cannot move out. It stays in. So we saw this on the previous slide. When water moves out, if solute doesn't follow, the volume goes down, but the saltiness goes up. So now, say it goes to 600, but now we have 600 being compared to 900. So more water is going to move out. So the volume is going down, but the saltiness is going up. So now it's 900 because the osmolarity in the tubular fluid increased. Well, now it's being compared to 1200. Water moves to the area of the higher solute concentration, so water moves out. And then, of course, this fluid is going to end up being 1200, where it is in perfect equilibrium with the interstitial fluid. So let's just review what happened. Water left, so the volume went down in the descending limb. So you say that you are reabsorbing water. You have to excuse me, my daughter just walked in the room, so she's sitting in my lap. So if you hear something, that will be her. So I think where we were talking about is, remember here, descending limb, the volume of fluid goes down because you're losing water, but since you're not losing salt, the osmolarity goes up. So this means in the descending limb, you are reabsorbing water because reabsorption is tubular fluid into the interstitial fluid. That's a good thing. It's good to reabsorb water. Well, now we can go back up the ascending limb. And remember what happens when we go up the ascending limb. Sodium and chloride is pumped out of the fluid. It's reabsorbed in the fluid, but water is not allowed to move. So that means the volume in the ascending limb remains unchanged, but the osmolarity now is going to decrease the higher up you go in the ascending limb because the solute is being pumped out. And eventually this goes up here, and I think it gets to about 150 or something like that, about 100, 150 osmolarity, because the volume's low, but now you have your little magic tweezers that you're pulling out the sodium and chloride. So the volume remains the same, but it loses its saltiness. And remember, the whole point of this is to reabsorb water. So here the fluid keeps traveling. Get my pen to work, there we go. The fluid keeps traveling, and now we get into the collecting duct. And this is really the importance of the nephron loop, which concentrates the medulla. Because without these levels of concentration, this deepening concentration, you're not going to be able to you're not going to be able to sufficiently concentrate your urine, which means you'll have to drink so much fluid just to maintain balance. But sometimes you need highly concentrated urine, and sometimes you need very dilute urine. And this is where the collecting duct is very sensitive to the hormone ADH. So ADH targets the cells of the collecting duct to open up. The presence of ADH opens up what are called aquaporins. So when ADH arrives, aquaporins open up. And so remember, here we go. We have this concentration of 150. If you have the aquaporins open, well, then you're going to lose water, or in this case, you're going to reabsorb water. So it's going to concentrate the fluid, but the further it moves down, you're going to keep reabsorbing the water. You're going to keep reabsorbing the water, and that's why you need that ever increasing concentration gradient so that you can keep reabsorbing the water. And this, in the presence of ADH, you would have a small volume of urine. When you do go to the bathroom, it's going to be a small volume of urine because you reabsorbed it, but it's going to be very yellow. It's going to be highly concentrated because the water was reabsorbed, but the solute remained the same. Of course, this is where the picture is going to get pretty messy. Of course, if you don't have ADH, these aquaporins are going to stay closed. The aquaporins are going to stay closed. And so then the fluid is just going to pass all the way through. So then you're going to have whatever the appropriate volume of the urine is, but it's going to be more dilute because you didn't reabsorb that water. So that is the function of the nephron loop. I do hope that this made sense. Look at the drawing that I have in Canvas, rewatch this. Hopefully now your book will make a little bit more sense. And of course, if you do have any questions, feel free to reach out to me, and I'm happy to go over this over Zoom or answer whatever I can in email.